Hello, in this video we will discuss about the Leishmaniasis. It is also known as the Kala-Azar or DumDum fever. It is a low degree fever can cause due to the Leishmania. So it is a vector-borne disease that is transmitted by sand flies and caused by obligate intracellular protozoa of the genus Leishmania. So the basically the human caused by more than 20 species, but also the animal also involve other mammals, and three main species involved for the infection is Leishmania mexicana and Leishmania amazonesis and the venezuelensis. So in this way, the identification of the pathogenicity is through basically the isoenzymes analysis and molecular methods as well as monoclonal antibodies. These steps we will discuss in the different video, but in this way you can just learn about isoenzymes analysis, molecular methods, and the monoclonal antibodies. This is the identification. So the identification status through are these three steps. But on the other hand, let's begin to understand the life cycle of the leishmania, the protozoa, which that is the eukaryotes because true nucleus containing protozoa. So remember, this require the vector. This vector is a sandfly stage, sandfly, and the sandfly stage we will discuss here when the sandfly will bite. So the sandfly takes a blood meal, injects promastigote stage into the skin. So the promastigote stage. So in this way here you can see this is the diagram of the basically the structure is the protozoa, which that will be inject into the blood circulatory system of the human. After the injection, the cycle will begin. In this way, the cycle we will discuss. The number one is the injection. The sandfly takes a blood meal, while the second one is the human stage. This human stage means the promastigotes are phagocytosed by macrophages in the human blood circulatory system or other type of mononuclear phagocytic cells like a granulocytes or the neutrophil or polymorphonuclear cell. So in this way, the phagocytosis will be occur of these promastigotes, and in this way, the promastigotes phagocytosed, and the third step is the promastigotes transform into amastigotes into the phagocytic stage, into the phagocyte, into the macrophage. After the fourth step is the amastigotes multiply in the cell of the various tissue and infected other cells also. And this is the diagnostic stage. We can diagnose it. So after the fifth stage is the sandy fly takes a blood again as a meal and ingest the macrophage infected with mastigotes into the sandy fly. And in this way, when sandy fly will be take the blood from the human, in this way the ingestion of the parasitized cell, which that will be the macrophage will be rupture, and this release this basically is the amastigotes will be released into the sandy fly, into the digestive system of the sandy fly. So the this is the sixth step. In this the ingestion of the parasitized cell, which that will lead to seventh step is the amastigotes transformed into promastigote stage in the gut. After the promastigote stage in the gut, basically the eighth step is the divided in the gut and migrate to proboscis of the sandy fly. After this, the divided in the gut and migrate to proboscis of the sandy fly. The again infection through a sandy fly takes a blood meal again and inject the promastigote, and the injection it will run again the cycle. And the infection we will discuss. First of all, we should need to understand the treatment. So the treatment is required. The drug is the pentavalent. We will also learn here is the presence in body, the liver, the bone marrow and spleen, which that is the major residential part of the residential place for this parasite. So let's begin to understand the treatment. The pentavalent antimonial SPV, the compounds including the sodium, sodium stibogluconate. This is the pentostem, or the traditional mainstays for treating the leishmaniasis since the 1940. While the we will let's begin to understand about the diseases. So this is the major diseases are present in the India. So the visceral leishmaniasis, it is also known as the Kala-Azar, and this basically the visceral leishmaniasis Kala-Azar. This is also known as a Kala-Azar or visceral leishmaniasis. This structure appear in the skin. This is a symptom, and in this way, the basically is the low degree of fever. The on the other hand, basically is the Kala-Azar remains a substantial public health problem in the country as it is present in 54 districts across 4 states: Bihar, Jharkhand, Uttar Pradesh, and the West Bengal, which that's contained here. Is you can see this is the district which that involve. But in this way, the low degree of fever, the hepatosplenomegaly means the swelling of the liver and the spleen, and cachexia and swollen lymph glands, as well as leukopenia, thrombocytopenia. The thrombocytes will be thrombocytopenia and monocytosis. The monocytosis means the monocytes will be decreased and loss of hair, and the fatal condition is the 90% of cases lethal and can cause death. So in this video we have discussed about the leishmaniasis, the Kala-Azar, and that is the video about the overview, so you can understand. And thanks for watching. Please make sure to subscribe, like and share if you like this video. So hopefully I will upload more about the parasitology, the video about the causing of disease through parasite, the eukaryotes. So thanks for watching.